So we have a tight schedule. So Brian, you are welcome to unmute. I'm going to get started right on time and keep moving right on time because that's what I've been instructed to do. So welcome to SRS Room 5. We now present Brian Gulley, who will speak on identity fusion to hometown as a predictor for pro-social community behavior. Please go ahead and put your questions in the chat to the host.
Thank you very much. I can share my screen and show my poster. Thank you, everybody. I'm presenting today for the Cox Lab on behalf of Brooklyn Kopp and Olivia Egloff. Our research is on identity fusion, which is a form of in-group identification that's a little bit more distinct than just regular group identification, in that it is personified by the willingness to act on behalf of a group, despite whatever self-sacrifice may come of that. And the literature on identity fusion in the past has been generally grim. It's often been linked with violent extremism, in that people who are strongly fused with, say, an extremist group or an ideology are willing to act violently or self-sacrificially and denigrate out-group members. But recently, the literature on identity fusion has taken a more positive turn to discover what pro-social implications can come from identity fusion. One study found that individuals who are fused to their nation were more likely to lend support to victims of things like the Boston bombing. So our research focused on one's hometown, defined as the city or town where one spent their formative years. This is a definition given to our participants. We were looking to see if identity fusion in town was of pro-social behavior. Right, we did that by asking 134 TCU students about their identity fusion to their hometown, and then asking them some questions about if they've ever considered running for public office, or if they tend to shop locally or vote in local elections. And our results indicated that this was the case. Individuals who were fused to their hometown were more likely to act in favor of that hometown, despite self-sacrificial implications like financial burden or just time burden. Fusion to one's hometown was also predicted of levels of kinship that one feels with other hometown members and their general well-being. And if I can move this here, the right side of our poster tells a little bit about the predictive ability of identity fusion. The top graph there, even though it's a little small, is related to people considering donating money to one's hometown in the event of an emergency, in the event of an emergency. And the lower graph, which you'll see is a little bit more powerful, pertains to how long individuals are willing to live in their hometown. So our regression indicates that the more fused someone is, the more likely they are to want to live in that hometown that they're from for longer periods of time. Now, in order to predict further implications, we're going to need a larger sample size. We're going to need to conduct more research of a more representative sample to examine how fusion and other predictors in one's hometown, like one's well-being, could affect their pro-social behavior in that hometown. All right. Thank you all for listening, and I'll take any questions.
Thank you so much, Brian.
Thank you. Stop sharing.
Do we have any questions from the audience?
Otherwise, I can just talk more about identity fusion as a concept. It's measured in a couple of different ways. There are intuitive measures of, like, using a pictorial scale where a small circle is representative of the self and a larger circle is representative of the group, which you can replace with any sort of group like a hometown. And the more in line they become is indicative of how fused one is. For our purposes in this study, we used a verbal identity fusion scale. So a general basic Likert scale with items like I feel at one with my hometown, or I'm strong because of my connection with my hometown.
That was a great presentation. Congratulations
So
on your
much.
research.
Thank you. Yeah. Thanks everyone for listening.
Absolutely. I don't have any questions now, so I think you're okay to sign off. Thank you so much for sharing with us. Our next presentation will start at 12:38. And then Carson, are you our next presenter? Great. If you want to go ahead and get set up and ready, we'll just keep moving really on time. We'll be very happy with us.
Can you all hear me?
Yes.
Okay. So for my CERT grant, I actually used it in relation with my honors thesis as well. And so I did my project and research on computer-assisted foreign language vocabulary instruction. And so to get started, I just want to go over some benefits. So benefits of learning a foreign language include, among others, enhanced employability, positive effects on cognitive functioning, and increased cultural awareness. Computer-assisted instruction can facilitate foreign language learning, and many computerized self-instruction programs are commercially available currently, such as Rosetta Stone and Duolingo. So the goal of our study was to compare the effects of two presentation formats when teaching foreign language vocabulary through computer-assisted instruction, and the high-density constructed response condition required a typed response in each trial. And then in the pair test condition, students viewed information passively, and typed responses were required only in intermittent test trials. And based on previous research on the effects of constructed response requirements in computer-based instruction, we predicted faster learning of new vocabulary in the high-density response construction rather than in the pair test condition, because better performance on translation post-tests and better 1 week retention of those high-density response construction, again, over the pair test words. 10 participants were recruited through SONA, and to participate, they had to report no previous knowledge of Arabic, and this was because we were teaching Arabic words in the study. The experiment was conducted remotely using the SuperLab 6 software and the remote control feature using Zoom, so participants were able to complete the study on the experimenter's computer remotely. Each participant learned the 10 Arabic words you see on the screen, and they included 2 fruit names, 2 animal names, and so on, as you can see on the screen. And 1 word from each pair was assigned to the high density response construction condition, and 1 to the pair test condition from each set. And the assignment of stimuli to conditions was counterbalanced across participants. So the learning phase of the study was divided into 4 training subphases and 4 label tests. Each training subphase had 40 trials, which is 20 for each condition. And the label test had 10 constructed response trials, 1 for each word. And then in each of the high density response construction conditions, the participant was shown a picture and asked to type the Arabic word in the first trial block, as you can see on the screen here. And for the next 2 trial blocks, only the first letters were shown on the picture, and they were asked to type the Arabic word. And in the first trial block, the word was shown on the screen, so the participant simply had to copy it, as you can see here. And then in the last trial block, the participant was not shown the word at all, as you can see here. And if the participant typed in the correct word, they were provided feedback saying that they were correct. And if they typed in the incorrect word, then the correct word was shown on the screen. For words in the pair test condition, the participant were simply shown a word paired with a picture, and if they did not need to respond except to advance to the next trial. And label test trials were identical to the other trials, except if the participants made an error, they were not shown the correct response. And so looking at the training phase results, the graph showed individual performance on the label test during the learning phase. And we use these data to look at the progress of learning, and filled circles show the high density response construction condition, and open circles show the paired test condition. So as you can see here, 5 participants had very similar acquisition curves in both conditions. And then 3 participants seemed to learn faster in the high density response construction condition. And then at the bottom, 2 seemed to learn faster in the paired test condition. And then on average, however, there was no difference between the 2 conditions. The translation post-test consisted of 30 constructed response trials without feedback, 3 for each word. 1 was a labeling trial that was identical to labeled test trials, except without feedback. And this was basically to just ensure that the participants would continue responding correctly when they were not given this feedback. And the other 2 trials were translation trials, where the participants were either presented with an Arabic word and asked to give its equivalent in English, or vice versa. On the post-test, again, there was no significant difference between conditions on any of the 3 types of trials. So the retention test was presented 1 week after the initial experiment, and it was presented on Qualtrics so that the participants could complete the test on their own time. And in the retention test, the participants were unable to recall many of the Arabic words in the label and native foreign trials. But once they saw the word in foreign native trials, as you can see on the graphs, they were often able to come up with the corresponding English words. But again, there was no significant difference between the 2 conditions. So in summary, we found no advantage of the high density response construction relative to paired test instruction when teaching foreign language vocabulary, which obviously shows that our hypothesis was not correct. But these results stand in contrast to the previous research that suggested the greater the density of constructed response trials, the more students learned. Possible reason for this is that past studies were taught more complex content than we did in this study, and paired test learning is less time consuming than typing a response in each trial. So practically speaking, the results that the paired test procedure may be a feasible way to conduct vocabulary instruction if time is of the essence. So thank you, and now I will open it up for questions.
Thank you so much, Carson. Do we have any questions from the audience? Carson, I wonder if you had to take a next step after this, and maybe you are already planning on that, but what would be your next step based on what you learned from this research?
That's an interesting question. So our lab is actually running I think an identical or very similar study that's actually being ran in Iceland right now. And I think they actually have ran a few participants and they actually got the same results, which is pretty interesting. As far as steps moving forward, though, I would probably think that teaching more complex like stimuli would be beneficial, because that's kind of where we got our hypothesis from, was from research that did more complex content as far as the stimuli are concerned. So I think maybe trying to teach some more complex words or stimuli would be interesting to look at, to see if those results from past studies could be replicated in a context like SONA. So seeing that would obviously be interesting.
Wonderful. Thank you. Do we have any other questions from the audience? You're welcome to unmute. We can help you unmute, or you can type them in the chat to the host. Okay, great presentation, Carson. If you want to stop sharing your screen. Congratulations on your research. Oh, we do have, I think we have one question actually. Let's see, there we go.
I just wanted to mention that the chat, I think, is disabled.
Oh, okay.
So that limits the engagement a little.
Thank you, Dustin. Are we able to correct that? Thank you, Matthew. Great job, Carson. All right. Our next presenter is Carly. Do we have Carly here? There she is. And Carly, I'll introduce your research. You can start getting all set up if you would like. And then we'll try and get that chat enabled. That way we can get some good questions for you. So next up, we have Carly Alley, who will speak on the effects of novel antioxidant treatment on microglial cell function in BV2 cells.
Can you guys see it?
Not yet.
Okay, let's try again.
There you go.
There we go. Alrighty, yes. So good afternoon, everyone. I'm Carly, and I'm actually going to be co-presenting with some of the people I worked with. So Paige, Caroline, and Margaret. We did a study the effects of novel antioxidant treatment on microglial cell function in BV2 cells. This was kind of the focus of my honors thesis kind of project as well. So kind of as an introduction, Alzheimer's disease, or AD as we'll refer to it as, is a progressive neurodegenerative disease that is the most common form of dementia. Clinical symptoms of AD include like memory loss and mental confusion. One of the key AD pathologies includes the extracellular amyloid beta plaques. The presence of these plaques endanger both inflammation and oxidative stress in the brain. And then therefore, with these pathologies, it's interconnected with the inflammation and the oxidative stress. So in collaboration with Dr. Green's lab in chemistry, they created a compound, L4, that has promising therapeutic properties for neurodegenerative diseases like AD. So prior studies have shown that AD, with the oxidative stress, L4 has properties against that antioxidant, and then you have oxidative stress in microglial cells, neuronal cells. So that was a previous study. Our study was the focus of the therapeutic properties of L4 against the inflammation response in BV2 microglial cells following stimulation from lipopolysaccharide, also known as LPS, which induces that inflammation in cells. And I think Caroline is going to be next, so she might need to be unmuted.
Hi, so for methods, we talked about microglial cells function like immune cells in the brain. So activated microglia rapidly respond to injury and infection by modulating inflammation in the central nervous system. So as you can see in this picture, when microglial cells are chronically activated by amyloid beta, they produce pro-inflammatory mediators and neurotoxic factors that result in neuronal damage and cell death. So in our current study, immortalized BV2 microglial cells were maintained in a cell incubator at 37 degrees Celsius and 5% CO2. So the cells were grown in complete cell medium, and when they became 80 to 90% confluent, they were passaged following our standard protocol. For the LPS treatment, BV2 cells were seeded into 6-well plates on day 1, and after 24 hours of growth, the BV2 cells were treated with various concentrations of LPS, and the cells were collected 4, 8, 12, 14, and 24 hours following the LPS treatment. For the combination L4 L4 pre-treatment and LPS treatment, BV2 cells were seeded into 6-well plates on day 1, and after 24 hours of growth, they were pre-treated with L4 at 5 different concentrations, and then treated with LPS at concentrations of 0.5 or 0.05 micrograms per milliliter 1 hour later. And then the cells were collected 8 hours following the LPS treatment. I think next up is Margaret. She will also need to be unmuted.
Okay, so I'm going to be talking about the results. After we collected the cell supernatant, we conducted enzyme-linked immunosorbent assays, also known as ELISAs, to measure the amount of TNF-alpha that was secreted by the microglial cells. TNF alpha is a pro-inflammatory cytokine released by activated microglia during an inflammatory response. So if you look at figure 5, it displays the TNF alpha ELISA results after treatment with LPS. Our results revealed that 8 hours of LPS treatment induced high levels of TNF alpha production in BV2 microglial cells. Also, 8 hours of LPS treatment with 2 different LPS concentrations were selected for use in future experiments with combined treatment of LPS and L4. And then if you look at figure 6, it shows the TNF alpha ELISA results after treatment with L4 and LPS together. The BV2 cells were pretreated with the L4 molecule in 6 different concentrations for 1 hour prior to LPS at 2 LPS concentrations stimulation for 8 hours. Preliminary data revealed that these particular concentrations of L4 pretreatment were likely not high enough to block the pro-inflammatory response induced by LPS. So in conclusion, although previous studies have shown that L4 has antioxidant properties that protect BV2 cells, our preliminary data revealed that we need higher treatment concentrations to protect cells from LPS stimulation. In the future, we will perform more cytokine assays with higher L4 concentrations and longer pretreatment time to assess the anti-inflammatory response of L4. So now we will be accepting any questions.
Thank you so much for that presentation. Do we have any questions from the audience? And if we can stop that screen share so you can see your audience. And the chat should be enabled for anyone who prefers to ask a question in the chat. It was a very smooth group presentation. Congratulations. Does your group have next steps in line where you're going to take this research?
Yes, so I guess I'll answer that one. So the next step, kind of as Margaret was mentioning towards the end, is we're gonna increase concentrations of L4, kind of pilot those higher concentrations, see where that takes us. Different ELISAs with different pro-inflammatory cytokines. Then we also have another pathway that's the antioxidant pathway of L4. So kind of look into that and just kind of run all the ELISAs, different types of assays like the MTT assay, and then just look into more proteins and enzymes with that. So yeah.
That's wonderful. Good luck to you in those next steps.
Thank you.
Thank you all very much. I believe next up we have Blake. And Blake, I will introduce you and your research, and then we'll get you started. You are welcome to start getting set up.
Can you hear me now?
Yes.
All right.
So we now present Blake Bazooka, who will speak on comparing different storm surge events and their effects on coastal vegetation, southern Louisiana. Please put your questions in the chat to the host.
Okay, let me share my screen. Okay. Can y'all see that?
Yes.
All right. So this is just a project I did for my GIS class. And my name is Blake Bazooka. And the title of the project is called the Katrina Storm Surge Estimates Using Elevation Models in Comparison to Landsat Imagery off the Louisiana Coast. And so I'm going to zoom to the background. So Louisiana coast obviously is prone to seasonal low pressure storm events that produce storm surges due to pressure differences as the storms approach the coastlines. So Hurricane Katrina is a great example of this. In some places it produced record storm surges of up to 9 meters in some areas. And so for this project, I'm going to use an average for my storm surge model of 7 feet, because that's what the average was for Hurricane Katrina. And so basically the objective is of the study is to determine if Landsat imagery can be interpolated or correlated to the amount of storm surge or superficial flooding that affected the Louisiana coast. So the data was collected from various sources. Landsat data was collected from the USGS website through the Earth Explorer portal. And then the DEM models were found on the ArcGIS online portal. And I also used various information from the internet for just general information. And so my methods that I use, so for the Landsat images, I tried to find some that were as close to the event as possible, Hurricane Katrina. But the satellites travel around the world every 2 or 3 days, and plus there's cloud cover, so it was hard to find. That was probably the hardest thing I had to do. But then once I got those and I got a clear image, I used just basically an NDVI tool on ArcGIS to create like a grayscale map that you see here, figure 1. And so basically dark values represent water. So Lake Pontchartrain is fully dark, and the darkest, but as you go on land, I guess I should say that the land values are white, or lighter values represent vegetation. And then as you go on land, you can see that there's actually some darker pixels in this image. And so I said that represented soil saturation, or storm surge, or flooding, or storm surge. And so the digital elevation model I used, basically I had the elevation model, the terrain model, and then I created a difference map. And that's what you see here of 7 feet. And so that's it on the topographic map around New Orleans. So some of the results I saw was that actually the DEM showed a good fit to the values of the NDVI. So here's on the bottom here is my my storm surge model of 7 feet. And then up here is my NDVI, and this is on the NDVI as well. And so I saw, especially in the middle, overall good fit to the data, but there's actually some other areas that I saw. So especially like right here, it overfit or I guess estimated the storm surge, and then over here actually underestimated the storm surge. So I thought that could be possibly because of maybe the type of vegetation that was there, or maybe just a variance in the storm surge that happened. Because like I said, the Louisiana or Katrina produced storm surges up to like 30 feet or 9 meters. Another thing I saw that was curious to me was that, so here's the Pascagoula River. It's one of the largest undammed rivers in the lower 48. And so I saw that there's an interesting interplay between just the drainage of the entire river going up dip compared to the storm surge down dip. And so I thought that this could be potentially a future study or something that somebody could use these types of data more screwed to be more, I guess, just to study this more, to see the interplay between river flooding, continental river flooding, and storm surges created by large storm events like Hurricane Katrina. So in conclusion, modeling storm surge with average values of surge due to Hurricane Katrina seemed to correlate well with NDVI soil saturation values gathered a few days after the storm itself, and differences in the data could be because of the variance in averages around the coast of storm surge, or even changes in type of vegetation. And so future studies could be considered after these types of events determine the interplay between continental river systems and actual storm surge on the coast. So that's all I got.
Thank you for that presentation, Blake.
I hope I didn't go over 5 minutes, but
it was great. You were right on time.
All right.
It was perfect. Do we have any questions from the audience? You can ask to unmute, or you can put them in the chat.
So yeah, it looks like we have a question from John Fanchi. He asks, how fast did the surge recede, and did your images see that average?
So that was one of the tough things I did. I think this was actually the clearest image. So there's actually some other areas that I thought were better, but there is just so much cloud cover, I guess, during this time on the Louisiana coast. So I don't know. I think I was thinking about that. I think maybe because I think it is representing soil saturation. So that could be it receded and that it just saturated, the soils are saturated there. But it also could be actual flooding. I don't know. That's something that I could look into later, or somebody else, because I have my own thesis to do. Yeah.
Thank you, Blake.
Yeah.
Any other questions? What brought about your interest in this particular topic?
So I just always found hurricanes interesting. So I thought this would be a cool thing to do. And in my thesis, I couldn't really do it. This project, I couldn't do a GIS project on my thesis because it's basically a geochronology study. So I had to find something. But I always thought this stuff was pretty cool, hurricanes, especially since, I have family on the coast. So they've always kind of affected me. So I thought it'd be cool to kind of learn about it some way.
Well, that's great. Congratulations, and thank you for that presentation. Do you want to stop your share? Great.
Okay.
All right. I think next up we have Ryan McKinney. And so, Ryan, you can start getting set up, and I'll give it just a minute, and then I'll introduce your research and you can get started.
Okay, thank you.
Okay. Next up, we have Ryan McKinney, who will speak on hydrogenation of reduced graphene oxide via water electrolysis. So please put your questions in the chat to the host.
Am I good to go?
Yes, please.
Is everyone able to hear me?
Yes.
Okay. So my name is Ryan McKinney, and I am in the Department of Physics and Astronomy. And this project was funded by the CERC grant, which is hydrogenation of reduced graphene oxide by water electrolysis. So to understand why this is important, we have to look at what hydrogen is. And that is its lightest element and the most abundant element in the universe. And this poses some problems with producing it, transporting it, and storing it, because it can be pretty dangerous if you put it in these compressed gas cylinders, as you see in the right, or if we cool it down and slow it down and turn it into a liquid, we put it in these cryogenic chambers. That uses a lot of energy, which is money, and that can still be dangerous, if somebody were to tamper that container at all. So these are the key challenges that you'd have to look at: storage and the transportation. So the material that I use is reduced graphene oxide. To understand that, we have to understand what graphene is. And graphene is a monomolecular layer of graphite, which when we oxidize it, becomes graphene oxide, which just has more oxygen functionalities. And if we reduce it, just kind of taking a step back, we actually reduce the amount of oxygen functionalities. So what I used was the reduced graphene oxide form. The hydrogenation part is simply a term used where we add hydrogen into it. And this can be in various interactions with the material, but the essence is that hydrogen is added. The initial goals of this project was to do electrophoretic deposition of electrochemically reduced graphene oxide, use these electrodes to perform water electrolysis, or separation of hydrogen and oxygen, and then analyze the storage yield of the hydrogen into our composite. So what's electrophoretic deposition, or EPD? It's using a power supply attached to two electrodes, one being a cathode where it has the negative potential, and one being the anode where it has the positive. Since reduced graphene oxide is a net negative particle, we want it to go towards the positive. So we attach the anode with our copper, which is the substrate that we use. When it undergoes this, it'll actually deposit onto the anode. This becomes electrochemically reduced graphene oxide. So this is the setup for finding the potential. We have the copper anode. We have hydrogen around the environment to keep any oxygen or other materials from getting involved in this reaction and messing it up. We have our counter platinum electrode, and then we have a sort of a referee a silver chloride reference electrode. This kind of just keeps everything predictable. And so here is the electrolytic cell that was one of many, actually. So here you can see the hose, you can see the electrodes, you can see the power supply. And what I found is that the mass of ERGO deposited on the copper versus time was fairly linear, not so much because there's a couple things at play: surface area, oxidation of the electrode, things like that. This wasn't a main focus, but it's good results to show. Here is the deposition of the chemically reduced graphene oxide, and it's a very thick layer. This is very important for storing hydrogen. What's another great thing is that we can actually take it off. And even when we take it off, we can look at the scanning electron microscope or SEM images, and we can find the presence of carbon, which is shown here. This shows that we actually deposited it even though we wiped it off. And so, all being said, we were able to do the electrophoretic deposition of graphene oxide onto copper, and to show that this could act as a hydrogen storage vessel. The ERGO could make storage and transportation, which is more economic and more viable for commercial use. And we were able to make various electrolytic cells with the funding. The geodeposition was confirmed, and our electrodes are reusable, which is another great thing for commercial and economic stability with this process. And the deposited ERGO can be removed from the electrode. These are good things to look forward in the future. So some of the future work that I would like to do and will be able to do is that the ERGO production will be corroborated. This is for publication means. And we can look at the carbon, hydrogen, and oxygen ratio of the hydrogenated ERGO to show an increase in mass percentage. And we can also look at scaling this up for possible commercial use. So if there are any questions, I will be happy to answer them, and feel free to ask them.
Thank you so much, Ryan. We have a question in the chat that said, great talk. Could you quantify the amount of hydrogen deposited?
So we weren't able to get to the exact step of hydrogenating it due to COVID and other aspects here. But typically what you would see in different attempts is about 6.5% biomass, meaning if you have hydrogen in there, 6.5% of the total mass will be of hydrogen. And so one work for this specifically was produced in 2019, and they found that the action of just depositing it was a 2% hydrogen by mass. So they didn't dry it and then reuse it as an electrode for hydrogenation. They simply deposited it and found 2%. So from this work here, we can say that there's at least 2%, but we could put more.
Thank you so much. Any other questions from the audience? I loved hearing that they could be reused. I think that's important in everything we all do, no matter our field.
Yeah, so it's got one more
Exactly.
question, and it's how do you recover the hydrogen for application?
So there's a couple, that's a good question. That's a good question. There's a couple methods. The main one is temperature at about 400 degrees Celsius, which is not very economically viable. You release the hydrogen, and that can be found. One attempt that I would like to try is applying electrical potential to the electrode with the stored hydrogen to see if that could actually release itself. In theory, this is a possibility. It's something we have to look at mathematically and see exactly what potential would be needed and how much will be released.
Thank you so much, Ryan. I think that is all the time we have for this one. To keep moving along, congratulations, great presentation.
Thank you, I really appreciate it. Thanks for the questions.
And if you'll unshare your screen,
You got it.
I believe we have Nam Nguyen up next. And so I'll introduce your research, but as soon as Ryan stops sharing, there you are. Oh, perfect, you're all set.
Yes.
So we now present Nam Nguyen, who will speak on evaluating the therapeutic efficacy of a small indole containing tetraaza matrocyclic pyridinophane for treatment of Alzheimer's disease. You are welcome to put your questions in the chat.
Hello, everyone. My name is Nam Nguyen, and I'm part of the Green Research Group, and I'm so excited to share my project with you today. So to first begin, when we consider neurodegenerative diseases, such as Alzheimer's disease, ROS, or reactive oxygen species, are often pinpointed as one of the main contributors to the development of these diseases. ROS includes species such as hydrogen peroxide, as well as the free oxygen radicals, the hydroxyl and superoxide radicals. Whenever ROS is misregulated within a body, it causes a plethora of biological damage. One of the ways in which ROS can be misregulated is via the misregulation of transition metal ions, such as copper-2. Whenever these transition metal ions are misregulated, it causes an extraneous generation of ROS. In the Green Research Group, we synthesize molecules to target diseases such as Alzheimer's disease. So we have to consider mechanisms of actions against things such as ROS. So to demonstrate that, I present a molecule we previously synthesized, OH-prion-3. It is known as a pyridinophane, meaning it's macrocyclic, large and cyclic, containing this pyridine group right here. We can see here how structure relates to function. So the nitrogens of the macrocycle are able to bind and stabilize transition metal ions when participating in ROS generation. Additionally, the OH group on the pyridine ring, known as the pyridol group, is able to contribute radical scavenging reactivity, meaning it is able to react with radical species and prevent them from causing damage within the body. So in this study, we study a new molecule known as indole pion-3 synthesized by group member Christoph Pata. And the reason we want to study this molecule is because it incorporates this indole moiety right here. The indole moiety is a powerful antioxidant, but most importantly, it is able to cross the blood-brain barrier, something that our ligands aren't able to do very well. So we want to see if the incorporation of the indole group onto the pyridinophane scaffold affects therapeutic efficacy. Will we still see radical scavenging reactivity? And additionally, will we still see the ability to halt ROS generation via transition metal ions? So to answer these questions, we first use the DPPH UV-Vis spectroscopic assay. DPPH is a free radical, and it models that of the free oxygen radicals that I previously spoke about. And we incubate our ligands with DPPH and see if DPPH is able to be quenched. So on the x-axis here, we have the concentration of the ligands that we tested, and on the y-axis, we have percent radical quench, quantifying the amount of DPPH left after incubation. And we test our ligand as well as a reference radical scavenger, BHT here. You can see that with increasing concentration, there is an increase in percent radical quench for both species, the reference radical scavenger and indole pion-3, answering our question that the indole group is still able to act as a radical scavenger when attached to the pyridinophane scaffold. So to answer our next question, we want to see if the indole ligand is able to stabilize transition metal ions participating in ROS generation. So how exactly does this work? When a transition metal ion, such as copper-2, is in the presence of a reducing agent, such as ascorbic acid, it undergoes redox cycling to produce ROS. We can take advantage of this and tracking this ROS generation by exposing it to a fluorescent molecule, in this case, CCA. When CCA is exposed to the hydroxyl radical, it readily converts to 7-hydroxy-CCA, which is incredibly fluorescent. So if you look at this graph, on the x-axis we have time, and on the y-axis we have RFUs, which quantify fluorescence. In this black series here, we have both ascorbic acid and copper-2, and you see a steady increase in fluorescence due to the formation of that 7-hydroxy-CCA and proving ROS generation. On the blue and orange series here, you have negative controls, which have either only copper or ascorbic acid, demonstrating you need both species in order to produce ROS. So we apply this experimental parameter to our ligand. So in half equivalents of the ligand, the yellow circular series, you see there's a decrease in fluorescence, but a much larger decrease in fluorescence with one equivalent of the ligand, demonstrated by this yellow square series here. Now we know from this study that our ligand is able to stabilize copper-2 and prevent ROS generation via redox cycling. So in conclusion, we now know that the incorporation of the indole group onto the pyridinophane scaffold does not affect therapeutic efficacy, warranting future study of this group in our ligand library. In future studies, we want to study other small molecules as well as evaluating other components of therapeutic efficacy. Thank you so much for your time. I'd love to take any questions at this point.
Thank you so much for a great presentation. Do we have any questions from the audience? You're welcome to put questions into the chat as well.
So it looks like we have one. They're asking, are there any side effects?
Are there any side effects? That's a great question. So that is definitely something that we would have to consider when we do later drug tests. There are many phases to drug tests. So right now with this ligand, to my knowledge, we have only tested what is called in vitro, meaning that we've only tested our ligand in non-living systems such as cells. So we would have to go step by step in a step-by-step fashion. So the next step would be to test this ligand in cells and then move on to like human trials, for example, and animal trials. And that is when we consider things such as side effects. But unfortunately, right now, we are just testing therapeutic efficacy in terms of in vitro. So side effects is not something that we can test as of right now, but it's definitely something we have to consider in the future.
Absolutely. Any other questions from the audience? What brought about your interest in this topic?
Thank you so much. That's a great question. I am a pre-med student, so I love to learn more about how we can contribute to health. And that's what drew me to Dr. Green's research group in the first place, is because there's a very medicinal aspect to this type of chemistry. And I just really enjoy being able to test. I'm more of a testing reacting molecules with each other rather than synthesis, which is why I was drawn to this kind of assay work. And that's kind of what drew me to this line of work in terms of research. And I'm loving it. Thank you for the question.
Well, it's fantastic. Thank you very much. And
thank you.
So that is all the time we have for that. Great presentation. And so next up, I think we have Emma. And so Emma, if you want to start getting all set up, I will introduce your research and then it'll be your turn to present.
All right, can everyone hear me and see my screen so far?
Yes, Emma, let me share with everyone. So this is Emma Kitchener, who will speak on multi-swabbing the deck, or swabbing the deck. And so if you will put your questions in the chat, she'll love to answer them at the end.
Hi, everyone. Yes, my name is Emma Kitchener, and I work in the spectroscopy fluorescence lab in the TCU physics department underneath Dr. Zygmunt Gryczynski. So today we will be talking about swabbing the deck. And specifically, we're going to be talking about the autofluorescence of swabs. So to be quite frank, the detection of DNA biomarkers is very important. It's used throughout all biomedical fields, and that should sound very familiar. And so it gets used anywhere from forensic science, using DNA to catch the bad guy, should we play Abby Shudo on NCIS, or maybe in a medical field testing for diseases or genetic mutations. But all of these tests have something very similar. They are all collected on medical collection swabs, and they all get sent for fluorescence testing, which is where I come in. So most of these swabs are in different sizes and materials, and most of them are used interchangeably without ever considering which one could be the best one for the job. And how we are looking at the best one for the job is which one has autofluorescence versus which one doesn't. So the autofluorescence that a swab could have, simply how bright the signal emission the swab itself gives off when excited by a certain wavelength, is purely due to the material the swab is made out of, and this signal can interfere with the detection of DNA. It can produce a false positive, meaning you think you have DNA, but you don't. Or if you know that it's there, it can actually hide DNA in its signal, so you have DNA, but all you see is the swab, so you're like, oh, there's nothing there. And both of those are just not good in the terms for detecting. So ultimately, my project is to detect minute amount of DNA on these collection swabs while battling the background signal. But my presentation today is purely just going to be looking at the idea of swabs having autofluorescence and what that information shows us, and if there's and just the inherent properties that a swab produces that we would then have to figure out how to combat later on. So for our experimental setup, we used our PicoQuant FT300 spectrofluorometer, and we adapted our system so that we could have our swab directly in the line of sight of both the monochromator as well as getting hit by the excitation. We 3D printed multiple pieces so that we can get our swab just so in an easy way. We then just ran this multitude of swabs. I just showed 4 for an example on figure 2, where when excited with 485 nanometers we get emission spectras of these. We then decided to select swab D because it looked kind of interesting to look at. This is known as the Salon medicinal swab. And we decided to run this swab both in dry and both wet with a PBS solvent to kind of mimic an environment that could be in the human body or in the fluids that we pick up with a swab. And so we ran a bunch of them, and as you can see, none of them look the same on initial look, and especially the emission intensity is crazy. So the next step to do was to normalize our emission intensity. And when we do that, we see that all of these curves happen to be the same. So what this tells us is, although one specific swab compared to the next one in this out of the same box might look very different, it's still made out of the same material, and it probably has just to do with how well it is wrapped around the stick that holds the swab. And so then we switched over and we looked at fluorescence lifetimes, which is essentially just the average decay time for an excited molecule. And we find that, and we compared it to a common dye that binds to DNA. And then that dye, when it is bound to DNA, which is what the blue and then green line are respectively. And we find that this red line, which is the swab, is the average lifetime of the swab, telling us it's shorter than the dye we would use, which sets us up for good conditions in future uses. Any questions?
Thank you so much, Emma, for a great presentation. Do we have any questions from the audience? You're welcome to type them in the chat as well. So Emma, where are you taking this now? What are your next steps in your research?
So we actually have already run through with a basic DNA binding dye. It's called an intercalator, aethinium bromide. And we've run through and actually figured out how small of amounts we can detect with the aethinium bromide and DNA combinations. And our next step is just to move to, we chose aethinium bromide because it's fairly well available and well accessible and cheap. Our next step would be to move into using official dyes from forensic fields, because that's actually the angle we're in particular looking to hit, and see how low of a concentration of DNA we can find using dyes that they use in their everyday field.
That's so interesting. Thank you so much. Any other questions? Okay, great, Emma. Well, you can stop sharing your screen, and congratulations on your research.
Thank you.
So we'll take just a minute. Our next up, I believe, is Jose Chavez. And Jose, you can start getting yourself all set up. At 1:25, I'll introduce you and your research, so you can get all set, and then we'll get you going here in just a second.
Okay, I'll just have my poster ready and I'll just leave it here for now.
That's perfect.
Yeah, so I'll wait 1 minute.
Yeah, make sure that those who want to come watch you talk, that we don't start too early.
Of course, of course.
Okay, so this is Jose Chavez, and he is going to speak on a study of direct excitation to the triplet state 5-bromoindole. And so if you all will please put questions for Jose in the chat.
All righty, thank you so much for the introduction. So again, this is my study over this poster session, which is called a direct excitation of the triplet state of fibromaindol. So for starters, I want to talk about real quick over tryptophan and how tryptophan is such a nice amino acid, other than that we must eat turkey in order to synthesize this specific amino acid, right? But one of the most important properties that tryptophan has is it is photoluminescent. So meaning it has something what we call fluorescence and phosphorescence. And the difference between the two is that the fluorescence has a typical lifetime of, say, a couple of nanoseconds, and that's roughly 1 billionth of a second. And then the other property that it too also has is called phosphorescence. And the phosphorescence is typically the afterglow effect that we would see whenever you shine a light at the material, and then you would immediately turn off that light. And then it has this glow in the dark effect where we would see something glowing that lasts in the range of microseconds to even to milliseconds, right? And so how the process starts off specifically, this is the intrinsic molecule that I would like to study, because typically in the fluorescence lab, we always would mark some part of the biological system that we're trying to study, right? Which is nice. But one of the main issues that we face whenever we're conducting some of these measurements is there's a lot of biological background noise. And so typically whenever you would shine a light at one of your probes, you would typically shine within the ultraviolet region, which will excite everything, including your probe. So all the garbage and your intensity measurements will be seen, and that's what typically we would like to eliminate. And so with tryptophan, this is one of the amino acids that is intrinsically bound to proteins, and this can be used to measure the protein misfolding. And there is some diseases that can come about whenever there's protein misfolding, such as Parkinson's or even Alzheimer's. And so this probe can be used to monitor some of these protein misfoldings in the time of the microseconds range. And so what we're going to be looking at here is we kind of want to use the long-lived lifetime of this tryptophan probe. And so the general theory of how fluorescence and phosphorescence has come about, the molecule will be absorbing a photon, and this is the process which is called absorption. And then one of two things can happen. The molecule can release a photon, and this is what we call fluorescence, or it can undergo a spin change state, and then this is where we call the triplet state, and then it too will release a photon, and it has a much longer lifetime, which is what we call phosphorescence. And so the idea that I would like to try out, or even try to explain, is that the process can be mitigated if we were to select not just an ultraviolet light to excite this intrinsic probe in protein. However, I want to choose a much higher wavelength in light, so that way it doesn't A, disturb the biological system that you're trying to study, and B, all of the junk that I was mentioning about, I want to see only the phosphorescence of this tryptophan probe, right? And so obviously we were studying the indole as shown here because this is connected to tryptophan, and specifically I wanted to look at fibromoindole because this bromide ion attached to indole will have an increase in probability in achieving the phosphorescent state. And so back in the around 40s or so, phosphorescence was still a thing that was studied within the scientific community. However, they were done in such low temperature to like even single digit Kelvin temperatures. And so what I kind of did in order to circumvent that is I used this polymer called PVA, and I've embedded the molecule inside in order to study its spectroscopic properties. So this polyvinyl alcohol will hinder the rotation of the molecule, and this will also possibly increase the probability in exciting to the phosphorescent state, right? And so we can see these nice pictures here. So this is the polyvinyl alcohol, then it gets polymerized, and then it gets embedded into the molecule. This is this drying drawer where I start my collection of films. And then finally, some of the results shown here. So this is what we call the absorption spectra, and this is typically how where you would shine your molecule with a specific wavelength of light. And over here on the right-hand side is the fluorescence emission spectra. So as we can see here with this black line is the indole structure all by itself, and it has a emission of about 325 nanometers. So that is something that we cannot see with the eye, but that's what the detector can see. And also the indole derivative as well. It too lines up with the indole structure, so that means they're pretty much the same. And then finally, we chosen this wavelength that excited at a much longer path, And typically this is shown here with the 2 excitations, first in the ultraviolet region. And then we found out that instead of exciting at 290 nanometers, it was actually at roughly 405 nanometers for indole. And so that's what we did. We checked with both the ultraviolet, so this is shown here in the red, and then the direct excitation of indole, which is shown in the black here. And these both fall within the phosphorescence range. So that means phosphorescence is indeed being achieved. And obviously, this is just the first step in my work. Hopefully, whenever I get a chance to continue on with this, I will be studying more of the different indole derivatives in order to study more of their spectroscopic properties. But I believe that's pretty much it. Are there any questions in the room?
Thank you so much, Jose. Any questions from the audience? You're welcome to put them in the chat as well.
Yeah, I can't see. Oh, no, I can't see the chat, so I might need help.
Oh, that's okay. I'll help you out.
Thank you.
Jose, what got you started on this line of research?
That's a very good question. So when I started in the lab, typically my advisor sees what we're pretty much good at. And so at the beginning, I liked studying spectra, and I kind of liked studying the theory of why, why certain things glow, right? And so my previous advisor, I did nanoparticles, and we studied fluorescence, right? And so I just love studying how the material interacts with light. And so as I joined to TCU, my advisor was like, you know what? Since you like spectra so much, why don't you try something different? You know, something that him and his brother did way back, but they never got a chance to even finish the work. So he's undergiven me this project, and it's been so interesting. And hopefully the findings that I get when I graduate, somebody will continue and possibly use this as a a technology to monitor protein misfolding, because I think this might be a good thing to start monitoring protein misfolding onto for Parkinson's and Alzheimer's. So yeah.
That's such an interesting concept. I can tell you're very passionate. It was
Thank you.
fun to hear you speak.
Mm-hmm.
Any last questions for Jose before we let him off the hook? Okay, thanks so much, Jose.
Thank you. I'll stop sharing. Yep. Okay.
Great. And so next up, I believe we have Corinne Laura.
Hi, how are you?
Hi, you can start getting all set up, and once you're ready, I'll introduce your research and then you'll be good to go. Okay. So we now present Corinne Laura, who will speak on individual differences in belief personification, moderate the effects of extrapolation on self-radicalization. And so please put your questions in the chat. Thanks so much.
So to begin, thank you all for coming and to listen about my research. Our study was to test individual differences in open-minded thinking, moderate the effects of extrapolation on self-radicalization. Our lab and I are very interested in self-radicalization, which we define as people adopting more negative attitudes towards a group or topic without ever learning new information about it. Past research has found that self-radicalization can occur when people simply think about a person or a group. Our lab has found additional evidence that suggests that certain types of thinking can also promote self-radicalization more than other types, such as when people extrapolate from known to unknown traits about a group. In a number of studies, our lab has found that people become self-radicalized towards target groups when they extrapolate beyond the positive or negative traits that are known to be associated with the group. And in the current study, we wanted to see whether people would report greater self-radicalization when they extrapolated about people who agreed and disagreed with them about a social topic. And we also wanted to see whether individual differences in as belief personification or judging people based on their opinions might moderate the effects of extrapolation and self-radicalization. For our methods in our study, we first asked participants if they favored or opposed kneeling for the national anthem. And our participants were then assigned to either an extrapolation or control condition. In our extrapolation condition, our participants read a cover letter about how the researchers were interested in the personality traits that characterize people who supported and opposed kneeling during the national anthem. Then they then typed the 5 personality traits that they associated with opponents of kneeling during the national anthem and 5 more personality traits that they associated with proponents of kneeling during the national anthem. In the control condition, the participants read a cover letter as well, but about how the research were interested in the personality traits that characterize different groups of people. They then typed into 10 empty text boxes synonyms that they could think of for 5 negative traits and 5 positive traits. Some examples of synonyms we gave were kind, controlling, ethical, intelligent, and some others. In our attitudes, next, all participants answered 4 items about their attitudes and behavioral intentions towards opponents and proponents of kneeling during the national anthem. I'm not going to go over the attitude measures individually for the sake of time, but I will present the combined attitudes in the results in just a few moments. The potential moderators, all participants, then completed a personality scale that is used to assess belief personification that was made by Lindman. And our results, as we see for attitudes, we found that extrapolators reported more positive overall attitudes towards people who agreed with them about kneeling and more negative overall attitudes towards people who disagreed with them about kneeling during the national anthem compared to the control condition, as we see here. And then in our individual differences on the bottom graph, the y-axis represents the attitude difference and refers to how far apart people were in their attitudes towards people who agreed and disagreed with them. And there was a greater attitude difference means greater self-radicalization. We found that self-radicalization was strongest for extrapolators who expressed greater belief personifications, while belief personification did not have an effect on the control condition. In our discussion, the current results are the first to assess individual difference in the effects of extrapolation on self-radicalization. And we hope in future research that we can explore whether the same findings would occur using different social or political topics. More research is also needed to determine other possible individual differences that might moderate these effects. We are super excited about the research that we're conducting, and we want to thank you all for letting me share it with you today. And if you have any questions, feel free to put them in the chat.
Thank you so much, Corinne. There's one question in the chat asking you what items were on the belief personification scale?
What items were on the belief personification scale? Sorry, my chat is not showing.
That's correct. Yes.
One second. We used we used the belief by Lindman. Some of the items people had to rate were included. Some phrases that we use to differentiate them there are a number of people I have come to hate because of the things that they stand for, or we use I tend to classify people as either for me or against me. Or another example we had was my blood boils over whenever a person stubbornly refuses to admit he's wrong. And we thought that these were good belief personification scales to really understand what side of the spectrum our participants were leaning towards in order to know if they were really extrapolating towards one side of the group.
Great answer. Thank you. What other questions from the audience? It's very timely research and very interesting. Okay, well, Corinne, I think you can stop sharing your screen. Congratulations on a great project and great presentation.
Thank you.
Next up, we have Claire. Claire, we'll wait just about another minute to get you started, but do you want to start switching over and getting your screen up? And then I'll introduce you and your research, and then you'll be free to get started in just a minute.
I think I might have to reset my Zoom really fast. For some reason, it's saying my system preferences needs to shut to be able to screen share. Can I do that really fast?
Sure.
Okay, sorry.
Zoom always picks the worst times.
I actually think it's working now. One second, sorry.
Great, okay. Well, you can.
Okay, is that working?
It is. Are you able to make it just a little bigger?
Yes.
Okay, perfect. While you do that, I will get you introduced. So everyone, this is Claire Clark, who will speak on the effects of extrapolation and trait imaginability on self-radicalization. And so if you will put your questions for Claire in the chat, she'll answer them at the end of her presentation. Go ahead, Claire.
Perfect. My study is about the effects of extrapolation and trait imageability on self-radicalization. In our lab, we're interested in attitude change, specifically in how people's attitudes about a group can become more extreme, even when they never learn any more information about that group. And we refer to this process as self-radicalization. Past research has found that self-radicalization can occur when simply thinking about an outgroup, but we suspect that certain types of thinking are more likely to promote self-radicalization than others. With this in mind, we've been looking at one type of thinking in particular, when people extrapolate from known to unknown properties about a group. And in a number of studies, our lab has found that self-radicalization towards outgroups can occur when a person extrapolates beyond negative traits they already know to be associated with an outgroup. For instance, sometimes we will tell participants that a group was observed demonstrating certain negative traits and then ask them to rate how likely it is that the group would also express additional negative traits. And in this study, we wanted to see whether the type of traits people extrapolated to mattered. And to test this, we manipulated whether it was easy or difficult to form a mental image of certain traits. We did this during an extrapolation task. And we actually initially predicted that participants extrapolating to easily imagined traits would report more self-radicalization than those with traits that were more difficult to imagine, but the opposite actually occurred. In this study, we first had our participants read a description about members of a fictitious migrant caravan. And then we told them that US citizens had lived with the caravan and described the people as suspicious, impulsive, strict, stubborn, dominating, opinionated, forceful, and possessive. Participants were then randomly assigned into one of three conditions. Those in the rehearsal condition were asked to retype the traits they were originally given about the group. The ones in the low imageability condition related how likely it was for the group to also have 11 moderately negative traits and 11 moderately positive traits, all of which were relatively difficult to form a mental image of. Lastly, the ones in the high imageability condition also rated how likely it was that the group would have the 11 moderately negative and positive traits, but instead all of these traits were relatively easy to form a mental image of. Then all participants reported their impressions of the group, attitudes towards admitting group members to the US, willingness to socialize with group members, and their willingness to do business with them. Before we get into the results, I wanted to point something out. The likelihood ratings during the extrapolation task were not at all affected by our imageability manipulation. Both the high and low imageability conditions reported that the negative traits were equally likely to occur and the positive traits were equally unlikely to occur. In terms of attitudes, both imageability conditions reported more overall negative impressions of the migrant group compared to the rehearsal condition, but there were no differences between the two imageability conditions. And in contrast, the results show that the low imageability condition was more opposed to admitting group members to the US, socializing with them, or less willing to socialize with them and less willing to do business with them. This is in comparison to the high imageability and rehearsal conditions, which there was no significant difference between on these three measures. The current results establish an initial link between self-radicalization and thinking about certain types of trait information during an extrapolation task. Our findings are actually the opposite of what we initially expected, and we think this might be because participants in the low imageability condition have to exert more cognitive effort to make sense of the traits that are difficult to form a mental image of. And because of this finding, we plan to, in the future, conduct more studies where we manipulate cognitive effort in different ways. And two possibilities for that are manipulating cognitive load or manipulating how easy it is to form a gist of the group information. We're really excited about this research, and I wanted to thank you all for letting me share it with you today.
Thank you so much, Claire. There is a question for you in the chat that asks, why do you think these effects occurred? What is it about extrapolating that makes people become self-radicalized?
Well, attitude theories suggest that attitudes are formed by the thoughts that are most salient at the time that people are evaluating something. And in this case, we think the act of extrapolation was what increased the salience of these negative traits while the participants were evaluating the outgroup. And it's also possible that the cognitive effort used to make sense of the extrapolated traits in the low imageability condition made them more salient than typically seen in our studies.
Great, thank you. What other questions from the audience? Claire, since we have a few minutes, what got you on this research line? What made you interested in this topic?
Well, honestly, I'm pre-med. I'm just a psych major. So I was looking at different labs and I was really interested in what Dr. Lord's lab is doing. And I just think it's really fascinating, like how different attitudes can change based on traits based on factors that don't exactly make sense sometimes. And it's just cool to start to understand why this happens to people. And self-radicalization is also really interesting to me.
Absolutely. Well, congratulations on great research. Last call for questions. Okay, well, Claire, you can unshare your screen.
Thank you.
Thank you so much. So up next, we have Michaela, and Michaela, we'll wait just a minute before you get started, but if you want to start getting set up, and then in just a minute, I'll introduce you and your research and we'll keep moving along.
Okay, sounds good.
Okay, our next presenter is Michaela Wilson, who will speak on new tricks with the Joker, revealing binary stars. If you all will put your questions for Michaela in the chat, she will answer them at the end of her presentation. Go ahead, Michaela.
Thank you for the introduction. Good afternoon, everyone. My name is Michaela Wilson. I'm a junior physics and astronomy major working with Dr. Peter Frenchboy, and today I'm excited to share with you my presentation, New Tricks with the Joker, Revealing Binary Stars. So binary systems, binary meaning two, are two stars that are gravitationally bound to each other, just like our Earth and Sun are. These binary systems can be found in various different ways. I have this GIF here that's showing that as the stars move in their orbits, the lines in their spectra at the bottom actually shift back and forth, and we call this Doppler shifting. So when we find these binary systems by looking at those Doppler shifts, we call them spectroscopic binaries. Spectroscopic binaries can then be further categorized as either single line or double line. So if one of the stars is shining much brighter than the other, we're not really going to see much information from that dimmer star. So we call it single line. And on the other hand, if we distinguish the lines of both stars in the spectra, we call it double line. And it's important to make this distinction because you can actually get more information about the stars when you're looking at a double line system. So these are two data sources that are capable of finding these binary systems. The first is the WIN Open Cluster Survey, which I'll refer to as WOCS. It's in the visible and it's also an older survey. And the second is the Apache Point Observatory Galactic Evolution Experiment. Kind of a mouthful. I'll refer to that as Apogee. And this one is in the infrared. So it can be kind of hard to characterize a binary system as double line when you're looking in the visible versus when you're looking in the infrared. And that's based on intensity ratios. You can actually see that difference in this spectral energy distribution that I have on the bottom right. So in other words, it's easier to see a system as double line in Apogee versus in WOCS. What's also useful to know is that the data from WOCS and Apogee overlaps in these 4 star clusters that I have listed. So this is exactly where we're looking for our research. So what am I doing? I'm using a software called the Joker, which is named after the well-known astronomer Johannes Kepler. And what the Joker does is it takes in the Doppler velocity measurements of binary stars and it plots out some possible fits or solutions to the data. So this figure, for example, is showing the Doppler velocity measurements of a star as the very small black points. And the gray lines are some potential solutions that the Joker gives. The Joker has actually already been run on Apogee data. So our goal is to input WOCS and Apogee together for a given star so that there's more points that the Joker can take into account when it's finding these solutions. As I mentioned before, WOCS is an older survey. So not only are we going to have more data points, the data can actually span over a longer period of time. And all of this will hopefully constrain the solution. So in other words, there's going to be less gray lines. These are some preliminary results that we've produced for a single star. The figure on the left is similar to that last figure, and it shows us the Doppler velocity measurements over time. The Apogee data is in red and the WOCS is in blue. And after we ran the Joker, we saw a really great initial fit to these points. So taking it a step further, we can actually fold this data over itself. And that gives us what's called a folded phase plot. And this is the orbital solution for the star. So this first test showed us that WOCS and Apogee data can be successfully combined. So that brings us to our future work. We're going to continue to run the Joker on the combined data for the 4 different star clusters in hopes of constraining these solutions. And we plan on using a second software that analyzes double line binaries. And what this is going to do is possibly reclassify some systems that we previously thought were single line as double line. And this is important because, as I mentioned at the beginning of the talk, double line systems give us more information about each star, like individual masses. And remember that we are also looking at spectra. So spectra are used to determine the chemical composition of stars. And this means that we have the potential to get a better understanding of the chemistry of the overall clusters. So I'll leave you with this SciComm statement that summarizes our research, and I'll be happy to take any questions that you have.
What questions from the audience are for Kayla? You're welcome to put them in the chat.
So it looks like we've got one, and it is how unique is the fit to the data?
Well, I can actually show you this image. This is from the paper on the Joker from its creator, Adrian Price Whelan. And you can see that there is a lot of solutions that it provides. It takes in some radial velocity or Doppler velocity measurements that are not necessarily high quality, but it's still able to produce some. So we get a lot, and that's why we are hoping to constrain the solutions by adding in more data.
What other questions from the audience? So I get to ask my favorite question. I love to hear how students came upon this research. What made you interested in this?
Yeah, well, my mentor, Dr. French Boy, I had him for classes and I just thought his research was really interesting. And usually for undergrads, they kind of just present you with the project and you take it on. But I was actually given the option of whether or not I wanted to learn the Yoker, learn what it's doing. And I took that project on because I really enjoyed the coding and software aspect of things. And I've really, really enjoyed it so far.
Good for you. I learned the new pronunciation. You can tell I'm not a physicist. Any other questions for Michaela?
Looks like one more came in, and
Great.
they're asking if you can automate the search to look at more clusters.
Well, we would have to actually choose the clusters that we're looking at since we're using both data sets. There's potentially more clusters that we could look at, but that's just based on whether or not the two surveys overlap and are looking at the same star clusters. So it wouldn't be applicable to just any star cluster.
Okay, great
Thank you for your
presentation.
questions.
Michaela, thank you so much. Congratulations on your research.
Thank you.
If you can stop sharing your screen, perfect. And I think for session one, our last up is Destiny. And Destiny, if you want to get all set up, and once you're ready, I will introduce you and your research, and then we'll be able to get started.
Okay. I'll share my screen.
Perfect.
Okay, so.
Okay, great. So we now present Destiny Longmire, who will speak on the effects of trait extrapolation on attitudes towards people who have similar and different opinions. And if the audience will put their questions for Destiny in the chat, she'll answer them at the end of her presentation. So go ahead, Destiny.
Okay, so my name is Destiny. The research that we decided to go with is called The Effects of Trait Extrapolation on Attitudes Towards People Who Have Similar and Different Opinions. So we are interested in self-radicalization, which is it's created as people form more negative attitudes towards a group or topic without ever learning new information about it. And the past research has found that self-radicalization can occur when people simply think about a person or a group. Our lab has also found that some additional evidence that suggests certain types of thinking, and these thinkings can promote self-radicalization more than others, such as when people extrapolate from known to unknown attitudes about a group. Something else we found in a number of our studies is that people become self-radicalized towards target groups when they extrapolate beyond the positive or negative traits that are known to be associated with the group. In the current study that we're going over today, we wanted to see whether people would report greater self-radicalization when they extrapolated about people who agreed and disagreed with them about a social topic, and the social topic that we picked was abortions. So in the method of our story, for our study, we asked the participants if they favored or opposed legalizing abortion, and participants were then assigned to either an extrapolation or a control condition. The participants in the extrapolation condition read a cover letter about how the researchers were interested in the personality traits that characterize people who support and oppose legalizing abortions.
Then they typed 5 personality traits that they associated with opponents of legalizing abortion and 5 personality traits that associated with proponents of legalizing abortions. In the control condition, we had participants read a cover letter about how the researchers were interested in personality traits that characterize different groups of people. Then they typed synonyms they could think of for 5 negative traits and also 5 positive traits. And then the attitudes, so this is how we got the results of the attitudes, is by all the participants answering 4 items about their impressions of proponents and opponents of abortion and their willingness to socialize with, do business with, and have their children, if they wanted children, to be taught by these taught history by the proponents of the opponents of abortion. So for our results, here in this top graph, we found that extrapolators reported more positive impressions of people who agreed with their stance on abortion compared to the control group. Extrapolators also reported a greater willingness to socialize with, do business with, and have their current or future children taught history by the people who agreed with them in the abortion stance. And in this bottom graph here, we found that extrapolators reported more negative impressions and less willingness to have their children taught history by people who disagreed with their stance about abortion compared to the control group. Okay, and then for this next part, the current results extend previous research and theory about self-generated attitude change and self-radicalization. And the future research should explore rather the same findings would occur using different social or political topics, and more research is also needed to determine whether individuals' differences might moderate the effects. So yes, this is what we did our research on, and I was glad to share it with you guys today.
Thank you so much, Destiny. There is a question in the chat for you. The question is, why do you think willingness to socialize with and do business with people who disagreed with the extrapolator were not significant?
Well, there could be a couple of explanations. Socializing and doing business with people who have different opinions may not be that important to people, especially in our type of society. So we are considering adding a perceived importance question in our future studies to determine whether or not people have perceived the topic as important for greater self-radicalization to occur.
Okay, great. What other questions from the audience for Destiny? I learned a lot on this topic today. Thank you so much for sharing your research.
No problem.
And I don't hear any other questions, then we will congratulate Destiny on a great presentation and great research.
Thank you.
And I would like to remind all of you, you'll probably hear this one other time, but the awards ceremony is at 4 p.m. and you will get to the awards ceremony using the link on srs.tcu.edu. And so the session 2 will start promptly, I believe, at 2:08. This is the end of session 1 for room 5. Thank you all for joining us.
Okay, thank you.
Thank you, Destiny. Good luck to you.